recording is about to start. It's wonderful. Let's put the screen share on. Start broadcast. Yep. And now let's go over to this one here. Can you see it? So all good, fantastic. And soon Moodle will open so that you can register your attendance. Please don't forget to do that. Yes, we can. That's exactly what I like to hear. That's Bob the Builder or Barack Obama. Attendance, fantastic. Thank you very much, guys. You are really good because it saves me from having to send the link. Wonderful. And I think everything is working and good, so that's excellent. So hello, everybody. Lovely to see you on a Friday, late afternoon, early evening. What do the points on attendance actually do? That's a very good question. We are, by law, we are required to monitor attendance because we have tier 4 students who are sponsored by the university. Their visa is sponsored by the university. And if they don't basically attend and engage, then their sponsorship can be withdrawn and they can be basically kicked out of university and be asked to leave the country. And because we can't single out individual students just because of their country of origin, we do the attendance monitoring for everybody. And if we find that students, for one reason or the other, don't attend classes, don't engage, don't do quizzes and things like that, then the academic advisor will check up on them, see if there's a problem, if there's anything that we can do. Is there anything we can support them with? Or are they just simply plain lazy? And if they are plain lazy, then we would be within our rights, if we can document that the students have not attended, then we would be within our rights to deregister those students. Yeah, I would agree with that. If you registered yourself absent, then I think, well, okay. Do we have another contributor to the pizza fund? Can you go back and register yourself as present? Oh, well, then just leave it. Don't worry too much. I will make a note. I mean, if people register being absent, then in a way, as Bernardo earlier said, it looks like they have been present, which is a weird concept. And I need to think about the philosophical implications of that, but let's not worry too much. No, 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 no. I don't think we can exclude the clowns from the pizza. So don't be mean. After all, they sponsor the pizza, so they are entitled to have a little bit, just a a slither of the pizza. It's amazing. The word pizza. Okay, let's get started, guys. Otherwise, I start salivating, and that's not a good thing. Um, okay, so most wonderful, good afternoon, early evening, ladies and gentlemen, and welcome to the BI300 Introduction to Biochemistry module. We will continue with the nucleic acids, and please register your attendance on Moodle. And people who log in, please make sure that your microphone is muted. That would be absolutely 100% appreciated. Only 3 quid. Oh, well, okay. So last time I virtually saw you, we were discussing DNA, and I told you the important features of DNA, that with DNA, we always have a double strand. So a double strand, it's the famous double helix. And I will try to write as neatly as I can, otherwise I get some complaints. So DNA is a double strand. It is the double helix. One helix goes from 5' to 3', and the prime numbers indicate the sugars indicate the carbons in the sugars of the strand, and the reverse complement strand goes from 5' to 3', but in the other direction, and we've got this helix. I told you that the base composition of a DNA is G, which has 3 hydrogen bonds that pair with a C, and also a C that pairs with 3 hydrogen bonds with a G. And we've got A that has 2 hydrogen bonds with a T, and the other way around. I told you that we can measure. No, I told you that we can isolate DNA by adding salt plus cold ethanol. The salt, in the salt, the positively charged ions neutralize the negatively charged phosphates and make the DNA less soluble. And cold and ethanol, again, makes DNA less soluble. So eventually the DNA will precipitate, or it will form some kind of sort of almost like a jelly-like mass. And next week, we are going to do a virtual practical with isolation of DNA from kiwi fruit. I think, did I already post the link to that? Is there a link for the virtual practical? Yes, I did. Luke says yes. Okay, good. So what I want you to do, actually, I think I posted it on the forum. But I will send you the link over the weekend again, and I want you to have a look at the practical and the exercises. So don't worry if you don't have it at the moment. So the positive, this is the positive ions in the salt neutralize the negative phosphate. That is what this abbreviation means. Sorry. So we did that. We talked a little bit about the reverse complement in a DNA strand, and that we always write from 5' to 3' end of a DNA sequence. So if we've got the sequence 5' ATGCA, for example, 3', that is the DNA sequence. Can you please give me the reverse complement strand of this sequence? Can you write down the reverse complements? Yeah, I see good answers. So we would have the reverse complement. You write it basically like T, G, C, A, T, 3', but we have to turn it around because we always write 5' first. So we would have 5', T, G, C, A, T, 3'. And most people got that. That's great. Please practice that because you will need this for the test next week. Oh, sorry, I shouldn't have said that. The other thing is that we need to figure out sometimes how many hydrogen bonds. And the reason why you flip it is because we always start with the 5' end when we write something. So how many hydrogen bonds can a T actually do? 2. How many are in a G? C has 3, A has 2, T has 2. So this sequence, this pentanucleotide, penta because it's 5, 1, 2, 3, 4, 5. So this can actually form 2 and 3 is 5, 8, 10. So there would be 12 hydrogen bonds in this molecule. And you might get asked how many hydrogen bonds are in a certain sequence. Okay, so we've done that. I also told you that in DNA, an absorbance in the photometer at 260 nanometer, at the wavelength of 260 nanometer, an absorbance of 1 is actually achieved by a concentration of 50 microgram per milliliter. So if you have a solution of DNA that has 50 microgram per milliliter, then it will give you an absorbance of 1.0. And obviously, if you have a different absorbance, or if you have a different concentration, you would have a different absorbance, but you always have to make sure that you measure less than 1.0, because otherwise the spectrophotometer can't deal with it. Now, Why do we see an absorbance? Why does DNA actually absorb electromagnetic radiation at 260 nanometer? Any ideas? Delocalized electrons, and they are the only ones that really absorb in this wavelength. It is the delocalized electrons in the bases, and the bases that we have are aromatic, and therefore they absorb the energy at 260 nanometers. Absolutely right. One thing is, because the electrons are so delocalized, what we find is that DNA does not just absorb exactly at 260 nanometers. In fact, so if we do an absorbance here, and here we have the wavelength in nanometer, what we see is something like that. The peak of DNA is at 260 nanometers. That's the peak. But there is also some absorbance going on, let's say, at 280 nanometers. Oh, there's another contribution to the Pizza Fund. I'm delighted to hear that. And it turns out that what we can do actually is quite clever, because pure DNA has an absorbance of roughly, when we have 50 microgram of DNA per milliliter, gives an absorbance of roughly 0.57 at 280 nanometers. So a little bit more than half of what we get at 260 nanometers. And what we will discover is that yes, King Chu, yes, that is actually the case. The electrons get excited to a slightly higher energy level. They jump up and absorb the energy that is delivered at 260 or 280 or any wavelength. So that's absolutely right. What we will discover is that at 280 nanometers, we also have other components when we have a crude mixture that are actually absorbing light. So what we can do from this spectrum, this is an absorbance spectrum, of pure DNA. What we can actually do is we can build a ratio. So we can, oops, where did it go? Don't know what I've done. We can actually do a ratio where we take the ratio of absorbance at 260 nanometer divided by the absorbance at 280 nanometer. And this spectrum is universally valid. It doesn't matter which organism it is. So as long as it is pure DNA, this spectrum holds true. And if you look at the wavelength, actually, we are in the UV range here, not in the infrared, we are in the UV range. So if I have pure DNA, 50 microgram per milliliter, what will be the ratio of 260 to 280 nanometers? Can you quickly do the calculation? 50 microgram per milliliter, what is the ratio? (260 to 280). Absolutely right. The ratio 50 microgram at 260 nanometer, that would be 1.0. That is what the absorbance would be at 260 nanometer, and 0.57, that is the absorbance at 280 nanometer. And if you put that into a computer or a calculator, you would get the A260 versus A280, as it is sometimes written. This ratio of roughly 1.8 indicates pure DNA without much contamination. Pretty pure. Not a lot contamination in it. If the ratio is below, so if A260 over A280 ratio is smaller than, say, 1.5, then this indicates that we have probably contamination of proteins. Contamination by proteins. So very easy to check whether you are contaminated, your DNA, with proteins. What you'd measure is you just take your DNA sample, and you measure it at two different concentrations. You measure it at 260 nanometer, and you measure it at 280 nanometer. You take the ratio of that. If it is around 1.8-ish, then you know you've got a good DNA sample. Okay, what about, somebody asked about 230 nanometer. Some people use 230 nanometer. This is usually not a terribly good indicator because a lot of substances actually start to do something in this 230 nanometer range. So you measure all sorts of weird and wonderful things, which is not a terribly good indicator. So in general, check two absorbances, two wavelengths for your DNA sample, build the ratio, and if it is around 1.8, it's good. Sometimes if it is much larger, So if the A260 over A280 is larger than 2.0, there's also a little bit, this is a bit iffy. There might be some other contaminants in it. Okay, so this is how you can measure the purity of DNA. For if we are working with RNA, and the rest of the session I want to spend on RNA. For RNA, you usually get a value of 260 for RNA, a value of 1.000 with RNA is about 40 micrograms per milliliter. But RNA also absorbs ultraviolet light. Why? Well, simply because it has nucleobases in it. Which are the nucleobases in RNA? UTCA. Oh, AUCG. That's absolutely right. So AUCG. These are the nucleobases in RNA, and in DNA, obviously, it's the T, which in RNA is replaced by uracil. Absolutely right. So if you've got an RNA sample of 40 micrograms per milliliter, you get an absorbance of 1.0 if you measure that at 260 nanometers. Okay, so what is the difference between RNA and DNA in general? As we just said, the base uracil replaces the thymine, and there are two other very important differences. Absolutely right. It's the ribose. That's one thing. So in DNA, our sugar, so here we have the 5' sugar, 1, 2, here's the 3' sugar. In RNA, we have an OH group, and in DNA, we have an OH group at the 3'. And in RNA, we also have an OH group in the 2' position, whereas in DNA, we just have a hydrogen in DNA. And there is a very important consequence of that, which people sometimes are not aware of. Let's just simply draw a sort of a strand. So we have sugar, carbon, we've got a phosphate here, that's the 5'. Here we have a base. Here we have O phosphate, and then we go into the next sugar. Next base here, O phosphate or phosphate, and we've got an OH group here, we've got an OH group here, right? So that is a typical RNA strand, right? Now, under the right conditions, what we can observe is that this OH group, which is very close to this O in the phosphate, this OH group can actually break this bond here. And it basically breaks the strand. So in a way, under the right conditions, and the conditions are alkaline pH, the RNA strand can easily break. And it is actually a hydrolysis reaction. You hydrolyze this strand. Water also plays a role, but it is initiated by this OH group, right? Can this happen in DNA? Absolutely not, because in DNA, you don't have an OH group there. That is why RNA actually is far less stable than DNA. DNA you can store for years, for 100 years, for 1,000 years. Actually, my wife is a molecular biologist, and about 20 years ago, it was 20 years ago, she worked on cloning some genes from, what did she do? It was genes from a leech, actually, which is incredibly useful. She was cloning some genes, and she really liked the genes. So she took a little bit of DNA, and the best way to store DNA is in a frozen state, because then it's not contaminated, or any bacterial contamination can't do anything. And we still have these DNA samples in our freezer here back at home. So there are these little DNA samples in our freezer, as you do. And the DNA is still absolutely fine. There is no damage. And no, we are not eating it. So absolutely right. At alkaline conditions, the OH group in the 2' position can cause strand damage. Keeping RNA alive is an absolute nightmare, and I probably will tell you a little bit about that at another time. Other people now said that RNA is also a single strand. So RNA single-stranded. And that's quite important. Remember, when I talked about DNA, I talked about base pairs, because we have a double helix. In RNA, we only have bases, no pairs. So RNA is always a single strand. Well, actually, I'm lying. because in most cases, RNA is not a single strand. RNA forms actually extensive, what is called secondary structures. And what on earth is a secondary structure? Well, imagine you have a DNA sequence, an RNA sequence, AUG, C, AUG, C, A, U, C, A, G, A, C, A, U. So here's the 5' end, and here would be the 3' end. And what you can imagine is that we can get base pairings here. Oh, here we don't get one. Here we don't get one. Oh, here we get one again. Here we get one. So what you see is we get base pairings with itself. And we can get very complicated structures with that. Very complex structures. So whenever people say it is a single strand RNA, yes, theoretically it is a single strand because it's not a double helix, but the RNA will fold back on itself. It will form areas that are heavily paired, and then you have sort of bulges where there's no pairing, and then you've got, again, pairing and things like that, again, where these where the strand folds back on itself, and rather complicated secondary structures appear. Does that make sense? Nope, they are not alpha helix or beta sheets. You find them only, these structures that we will discuss when we are dealing with proteins. We are usually talking about stems in terms of RNA, stems and loops and bulges when we are talking about RNA. Now, let's have a look at what species of RNA we could actually encounter. And for that, I want to quickly go with you through the central dogma. It's just called stems, loops, and bulges, these secondary structures. So, at the beginning of the central dogma is the DNA. This is the carrier of the information. And the DNA, of course, is replicated and synthesized, but that is the guardian of the information. If we want to do something with the information of the DNA, what do we produce? What is it that we produce? Absolutely right. We produce what is called mRNA, where the M stands for messenger. The messenger RNA carries the information of the DNA. This is the process of transcription that we have here. And before we actually arrive at the mature mRNA, we have what is called the hnRNA, heteronuclear RNA, or pre-mRNA, pretty much similar things. And through a lot of modification of the pre-mRNA, at least in eukaryotic cells, in these cells, we then eventually get the mRNA. For example, splicing, we can add a cap to the one end of the RNA, we can add poly A tail at the other end. So there is lots of modification going on in the mRNA, but eventually we end up with the mRNA. The mRNA is then transported out of the nucleus where everything happens and to the ribosomes. And in the ribosomes, we produce the proteins. And of course, this process is called, what is this process called? That's the translation, exactly. That's translation. So please don't mix these two things up. Transcription, DNA to RNA, to messenger RNA. Translation, mRNA to protein. We still have second-year students who get confused by these two things. It's very different processes. And proteins then also do extensive folding, and we will discuss that as well, to get into the right shape. Now, so you've seen this scheme. Big question for you. How much of the RNA that we find in the cell, how much RNA, let me write this properly. How much RNA in the cell, RNA in the cell is actually coding RNA. With coding RNA, I mean mRNA or hnRNA. How much do you think of the RNA in the cell is coding RNA? 20%? 99%? 5%? Interesting. Actually, it turns out that it is around, probably around, say, 10% of the RNA is coding RNA. Only 10%. So what the heck is all the other RNA doing? All the other RNA is non-coding RNA. Non-coding RNA. And what is non-coding RNA? Well, non-coding RNA is all those RNA molecules that are not mRNA. So not mRNA molecules or hnRNA. So they are not mRNA. The biggest bulk of them is ribosomal RNA. Ribosomal RNA, abbreviated as rRNA. So hnRNA is heteronuclear RNA. It's just big RNA that has not been processed. And that is before it becomes mRNA, exactly. Ribosomal RNA, that's about 80% of all the RNA in the cell. And the ribosome, as we know and love it, is predominantly made up of this rRNA, of rRNA molecules. We also have what is called the transfer RNA, and we will discuss transfer RNAs in more detail. They have a very characteristic structure and function. So what a tRNA molecule looks like is it has what is called the cloverleaf structure. Here is the 3' end. So we've got lots and lots of base pairs or pairing here and here. So it has this very characteristic cloverleaf. Here we've got the 3' end. The 3' end actually has a characteristic sequence C, C, A. And this A, we can attach an amino acid. Amino acid. And these tRNAs are actually delivering amino acids to the ribosomes where then a protein can be translated. We also have here this region here, and I draw this larger. This region here is called the Anticodon. Anticodon loop. Absolutely right. The tRNA has to get into the ribosome to deliver the amino acid with which it is loaded. So here we have the anticodon loop. So we are here still with the 5', and here's the 3' part of this loop. And for example, we have 3 bases here, which are called the anticodon in the tRNA. And you will hear more about the loading of the tRNAs with the amino acids in the BI300 module, how this actually works. So we've got anticodon. These are 3 bases here. And the easiest way to illustrate what they do is if we take some mRNA, so mRNA, let's say we have the sequence 5' A, 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 A. That's a really boring sequence, but just to illustrate that. Now that is the mRNA. And in the process of the translation, we have to add the right amino acid. So we have to get the right tRNA in there, and that it is the right tRNA. It will form base pairs with A's. What does pair with A in RNA? Yep, it's U. So we have, how shall I do that? Yeah, let's do it like that. U, U, U. So this is our anticodon loop here. This is the 5' end. So here's the 5' end. And here would be the 3' end, right? Does that make sense? So I draw it so that the U's actually, they are standing on their heads, so that the U's can pair with the A's. How many hydrogen bonds do we have? 2. Well, yeah, so we have got 2 each in this case. And we are always looking at just one triplet at 3 bases. Okay? So what we see here is we've got our 5' end here. So in the 5' end, basically the first base here, this one here, that's the first base in the anticodon loop, is the 3rd base in the mRNA loop. Yeah, that's this guy here. Let me draw them in a different color. This base here is this base here, and it is the 3rd base in here. And what you find sometimes, and I will post a video on that, is that sometimes the first 2 of these are fixed. So we have to have A pairs with U and G pairs with C. That's the first 2 that are fixed. They cannot do anything else. In the 3rd position, that's the 3rd position in the mRNA, 3rd position, we have a little bit of flexibility. No, you don't need to remember which codon codes for which amino acid. Don't worry about that. The 3rd position here, we have flexibility. Flexibility. Yes. So in the 3rd position, what can happen is that sometimes we can pair G with a U, or we can even pair G with an A. And that really stunned people, but it is generally known as, does anyone know what this is called when it sort of has this flexibility? But they are not complementary, absolutely right. But they can, if forced, they can do some sort of deal. It's not semiflex, it's called Wobble. And we can discuss that when we are discussing the genetic code next week. So there is a Wobble element in it. Does that make sense? And there is a biological significance to it, why we have this flexibility in a way. So we have non-Watson-Crick base pairing, sometimes in the 3rd position. Now somebody said it's called Wobble. Yes, I'm terribly sorry. Why don't they do that anyway? Because it would be dangerous to do a Wobble in the first 2 positions. When we discuss the genetic code next week, we need to be absolutely fixed with that. And somebody said the code is redundant. We will discuss that. And hopefully I will convince you that the latest research probably shows that the code is not redundant. But I don't want to spoil the fun. That's what we are going to do next week. We are talking about the genetic code and the amino acids, the function of amino acids. And we are also going to have an extended session on Wednesday about the kiwi DNA extraction practical. School lied to you. School lied to you in two aspects. School told you that the genetic code is redundant and the genetic code is universal. And the genetic code is not universal. And I can unfortunately tell you a story about that. How I got stung by the genetic code not being universal. You want to know why it's not universal? Is that right? You want to know why? Stay tuned. I tell you next week. Have a lovely weekend, and I will send out a checklist and things like that over the weekend. So thank you very much for watching, and enjoy your weekend. Take care. Bye-bye.
Hi, sir. Are you still there?
Wimmy.
Oh my god!
Wimmy.
You scared the shit out of me.
Have you got a lecture?
I just